two-dimensional cell cultures do not adequately mimic in vivo tumor growth. To improve, 3D procedures have been developed using spheroids. Our 3D glioblastoma spheroid-based assay is particularly well suited to investigate brain tumor invasion into the healthy brain tumor environment.
Three-dimensional culture models can be used to recapitulate the multicellular architecture, heterogeneity, and metabolic state of tumors, allowing a more rigorous evaluation of drug effects.
Be sure not to touch the bottom of the well or to completely remove the supernatant, to keep the gel on ice, and to add the cells to the gel quickly.
To generate uniformly sized tumor spheroids, first wash the tumor cell culture of interest with 5 milliliters of PBS and treat the cells with 0.5 to 1 milliliter of dissociation enzyme. After 5 minutes at 37 degrees Celsius, stop the reaction with 4 to 4.5 milliliters of PBS and transfer the detached cells into a 15 milliliter conical tube containing 10 milliliters of complete growth medium. After counting, resuspend the cells at a 1 × 10 to the 6 cells per 8 milliliters of complete growth medium supplemented with 2 milliliters of 2% methyl cellulose concentration, and transfer the suspension to a sterile system container. Then add 100 microliters.